Hi, I'm Mark LaBarge. I'm a professor from the City of Hope in the Population Sciences Department, and my lab studies breast cancer in the context of aging, trying to understand why, as women get older, they get more susceptible to breast cancer. So we've been building systems for the last 15 to 20 years that let us study human cells in a human tissue-like context. What I'm going to talk about today is a new technology that we've invented called microcontainers, which is an organoid-based technology that allows use of recombinant organoids, stem cell-based organoids, and just tissue fragments. And we can look at them in a very statistically robust format. We can do it in the absence of exogenous matrices from mice or synthetic. And we can perform these experiments over a very long period of time. We're almost out to a year currently in my lab, watching individual organoids over time. So as you may know, organoids are an attempt for us to bridge what's happening in vivo in an in vitro system. And particularly, this is important in humans because we're difficult to work on. We like our tissues, and there's a lot of ethical issues involved in working in humans. So there's three general flavors of organoids, either stem cell derived, derived from chunks of tissue directly, or recombinant organoids. And the system we've developed works for all three of these different types of organoids. There's been, for many years now, attempts at making various organoid systems. Embedded and on top matrigel type cultures are the most popular. We've worked a lot with microwells, which are sort of an open top, small, confined volume spaces. And then there's other things like hanging droplets and sphere type cultures. These all lack they make it difficult to do statistically rigorous experimental design. And importantly, all of them either completely lack an extracellular matrix, which is key to normal cell function in vivo, or they have to use a recombinant or synthetic matrix. And our system, the microcontainer, solves all of these. So the microcontainer is basically based on a microlithography technique where we make microwells. We load cells into the microwells. We add a dense solution of maltodextran and then flow over that a lid. And one of the surprising outcomes of this is that we were able to achieve culture in a normal organization, normal polarity of these tissues in the absence of any matrigel or any other synthetic culture. And there's a slide on screen now that sort of shows you what that basic technique looks like. We can individually address up to 7,200 of these organoids and watch them for a very long period of time. And as you'll see on this little power analysis off to the right side of the screen, there's very strong statistical power built into the system. We started from thinking about statistical design. Most organoid systems completely lack this element. And so this is actually very important. And as you'll appreciate, a lot of things that happen in tissue happen in subtle variations. And what we're looking for are those subtle changes that are important to human health. And we can detect 5 and 10% differences. We can do this over very long periods of time. And so we've gone out almost to a year at this point. What's on screen now is looking at lumen formation in mammary epithelial organoids. They look more like acini in this particular representation, and this is going out 45 days. But using machine learning algorithms to track each individual acinus, we can get very strong time lapse and sort of longitudinal data on behavior of cells and tissues. So what you see on screen now is an example of the pretty amazing fully human matrix. So these cells, because we provide a constrained geometric volume, we think that they're able to secrete and provide their own native matrix. So down here on the lower left-hand corner, you see we've actually used tweezers to pull out plugs that were created by the cells themselves. So this is where the fully human matrix is coming from. And just to the right of that are some immunofluorescence images where what you're seeing are the creation of matrices that are impossible to create in a mouse system. So these could only have come from a human cell type. We always try to compare what we're doing back to in vivo. So my lab has been focused mainly on breast, and we've done a lot of this development in breast. So what you see on the top left are cross-sections of actual breast in humans, and below that are the same bilayered organization and branching that we get in our lab-based organoids using the microcontainer system. We've actually achieved heretofore unachieved levels of functional differentiation, such as contraction and lactation using this organoid system. And again, to just really highlight the strength of the robust design here, you have, you can separate fixed effects from random effects using this cell culture design system, where we're using basically a 24-well plate. In each 24-well, there's 300 organoids. So that's 7,200 organoids. So you can look at 24 different conditions and various combinations thereof, depending on your statistical requirements. In conclusion, we're looking for partners who would like to either directly commercialize the technology as kits, looking for people who might be interested in developing an SBIR grant together, or looking for companies that might like to deploy this in a preclinical setting to further their own drug development and research. And if you'd like to take any next steps or talk to me further about this, please feel free to reach out by email. Thank you.